Immunology Insights, Basics and Beyond. Chapter 12, Antibodies and their Subtypes. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored the pioneering work of Elie Metchnikoff and his discovery of phagocytosis. Today, we shift our focus to the molecules that are central to the immune system's ability to recognize and neutralize pathogens, the antibodies. Specifically, we will delve into the different types of antibodies, IgA, IgD, IgE, IgG, and IgM, and their unique roles in the immune response. So, let's talk science. To introduce antibodies. Well, antibodies, also known as immunoglobulins or Ig, are Y-shaped proteins produced by B cells and play a pivotal role in the adaptive immune response. These remarkable molecules are designed to recognize and bind to specific antigens, foreign molecules on the surface of pathogens with extraordinary precision, much like a lock and key. The generation of antibodies is a sophisticated process involving multiple layers of molecular and genetic mechanisms that ensure their specificity and adaptability. B cells, the cellular factories of antibodies, originate in the bone marrow. During their development, they undergo a unique process called VDJ recombination, where segments of antibody genes, variable, diversity, and joining segments are shuffled to create an immense diversity of potential antigen binding sites. This diversity is further enhanced by somatic hypermutation, a mechanism that introduces targeted mutations in the antibody's genes during the immune responses. These processes are central to the generation of antibodies with a high affinity for specific antigens, a phenomenon fine-tuned in specialized structures known as germinal centers within lymph nodes and the spleen. An antibody consists of two main regions, the Fab region, which binds the antigen, and the Fc region, which interacts with other components of the immune system, such as complement proteins or immune cells. This molecular structure enables antibodies to both recognize threats and recruit other parts of the immune system to eliminate them. From an evolutionary perspective, antibodies are an ingenious solution that has developed in vertebrates to provide targeted and adaptable immunity. Their genetic underpinnings, involving constant and variable regions, allow for both stability and flexibility in the immune responses. The five main classes, IgA, IgD, IgE, IgG, and IgM, have evolved distinct functions to combat pathogens in different contexts, from mucosal surfaces up to the bloodstream, showcasing the immune system's versatility. Thus, antibodies are not only a cornerstone of immunological defense, but also an evolutionary marvel, demonstrating the interplay of genetic mechanisms, cellular processes, and molecular precision in safeguarding our health. Now, let's come to the different subtypes of antibodies. Let's start with IgA, the mucosal defender. IgA is primarily found in mucosal areas, such as the respiratory and gastrointestinal tracts, as well as in bodily secretions like saliva, tears, and breast milk. Its primary role is to protect these vulnerable entry points from pathogens by neutralizing them before they even can enter the body. IgA exists in two forms: monomeric IgA, found in the blood, and dimeric IgA, which is the predominant form in mucosal secretions. Dimeric IgA is linked by a secretory component that protects it from being degraded by enzymes in mucosal surfaces, ensuring it remains effective in defending against pathogens like bacteria and viruses. One of the most significant roles of IgA is in the neonatal immune system. Breast milk, rich in IgA, provides infants with passive immunity, protecting them from infections during the early months of life when their immune system is still developing. Next, we have IgD, the enigmatic immunoglobulin. IgD is one of the least understood antibodies, and for a long time its function remained elusive. IgD is primarily found on the surface of immature B cells, where it acts as a receptor for antigens. This role is crucial in the early stages of the immune response, helping B cells recognize antigens and become activated. While IgD is present in a very low level also in the blood, it plays a significant role in the respiratory immune system. Recent research suggests that IgD may help in the defense against respiratory pathogens by interacting with basophils and mast cells, triggering the release of antimicrobial factors. Despite its enigmatic nature, IgD is essential for the proper functioning of the immune system, particularly in the initial activation and regulation of B cells. Then we have IgE, the allergy mediator. IgE is best known for its role in allergic reactions. It is found in very low concentrations in the blood, but plays a critical role in the immune system's defense against parasitic infections such as those caused by helminths, which are worms. IgE binds to allergens, substances that cause allergic reactions, such as pollen, dust, mites, and certain foods. Once bound, IgE interacts with receptors on the surface of the mast cells and basophils, triggering the release of histamine and other inflammatory mediators. This release leads to the symptoms of an allergic reaction such as itching, swelling, and in severe cases, anaphylaxis. In addition to its role in allergies, IgE is crucial in the defense against parasites. By binding to parasites, IgE helps to coordinate the immune response, recruiting other immune cells to attack and eliminate the invaders. Next, we have IgG, the versatile defender. IgG is the most abundant antibody in the blood and extracellular fluid, making up about 75% of the total immunoglobulin pool. It is the primary antibody involved in the body's long-term defense against pathogens. IgG is highly versatile, capable of neutralizing toxins, viruses, and bacteria. It can also activate the complement system, a series of proteins that work together to lyse, which means break down, pathogens and enhance phagocytosis by macrophages and neutrophils. IgG is the only antibody that can cross the placenta, providing passive immunity to the fetus. This maternal IgG protects newborns during the first month of life, until their immune system becomes fully functional. This is complementary to the role of IgE in maternal breast milk. There are 4 subclasses of IgG: IgG1, IgG2, IgG3, and IgG4, each with slightly different roles in the immune response. For example, IgG1 and IgG3 are particularly effective at neutralizing viruses, while IgG2 is more involved in the response to bacterial polysaccharides. Last but not least, we have IgM, the first responder. IgM is the first antibody produced by the immune system in response to an infection. It is predominantly found in the blood and lymphatic fluid and is the largest antibody, forming a pentamer structure with 5 antibodies units joined together. Due to its large size, IgM is highly effective in agglutinating, which means clumping together, pathogens, which helps to prevent their spread and makes them easier targets for phagocytosis. IgM is also very efficient at activating the complement system, leading to the rapid destruction of pathogens. Although IgM is the first antibody to respond to an infection, it is gradually replaced by IgG as the immune response progresses. However, IgM remains crucial for providing early defense against pathogens, especially during the initial stages of an infection. Of course, there are also therapeutic and practical applications of antibodies in medicine and science. Beyond their natural role in immunity, antibodies have revolutionized medicine and science through their therapeutic and diagnostic applications. Monoclonal antibodies are widely used as treatments for diseases like cancer, autoimmune disorders, and infectious diseases. these engineered antibodies can target cancer cells directly, block harmful immune responses in autoimmune conditions, or neutralize specific pathogens. In molecular biology, antibodies serve as indispensable tools. Techniques like flow cytometry rely on fluorescently tagged antibodies to analyze cell populations, while in ELISA, which means enzyme-linked immunosorbent assay, antibodies are used to detect and quantify specific proteins, such as biomarkers in diagnostic tests. Antibodies are also essential in Western blotting, immunohistochemistry, and other techniques pivotal to scientific research. While antibodies are essential for immune defense, they can sometimes also contribute to diseases. In autoimmune diseases like lupus or rheumatoid arthritis, the immune system mistakenly produces antibodies against the body's own tissues, causing inflammation and damage. As also described before, allergic conditions are another example where IgE antibodies drive exaggerated immune responses to harmless substances like pollen or food. Understanding these antibody-associated diseases not only sheds light on immune dysfunction but also opens avenues for targeted therapies. To conclude, antibodies are central to the immune system's ability to recognize and neutralize a wide range of pathogens. Each type of antibody, IgA, IgD, IgE, IgG, and IgM, plays a unique and vital role in tailoring the immune response to different threats. Understanding these differences helps us appreciate the complexity and precision of our immune system. In the next chapter, we will explore the roles of cytokines and chemokines, the signaling molecules that orchestrate the immune response, ensuring that all parts of the immune system work together in harmony. Stay with us as we continue to uncover the intricate workings of the immune system. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.